All right, let me do the beginning. Thanks for joining our session today. I'm very happy that at the last moment on Monday, we got legal approval by our customer Interven. And so we have Karina with us, who's basically presenting the rest of the presentation. I'm really happy to have her here. Karina, can you go to the next slide? I'm just setting the stage here. So what are you going to see in the next 15 minutes? We talk about glycan structures, which is basically sugars that are connected covalently, so like in chemistry and linked with each other. And you see some examples there on the right side, these little trees there. So basically, yeah, they are graphs. And the glycan composition, so you see the colors and the shapes of the different sugar monomers are connected to each other in a tree-like structure. And depending on the composition and the length of these sugar trees, they are disease-specific, for example, for Alzheimer's disease or for cancer. And that makes them really interesting to study them in terms of targets. And Karina, if you go one slide back, we got the problem that the methodology of measuring that in the lab is not really 100% accurate. And that need some more technical, let's say, supporting material. And because we have kind of pattern matching problem and in a graph, it makes us a very, very cool use case for graph technology. And so on the lower one, you see like sometimes we cannot see what's in between this molecule there with these question marks, so we don't know how long the sugar tree is. And then on the right side, you see just a mirror image. Sometimes we don't know the orientation of the monomer sugar molecule. Now, Karina, go to the next slide. That brings me to basically the first conversation that I had with Karina, and we draw kind of a little whiteboard session here, how we can do that in Neo4j and using that with variable length queries. And so this is basically the handover to Karina, and she will explain you how far they get so far. So Karina, stage is yours.
Yes. Thank you, Alex. That's very kind introduction. So I'm very happy actually to be here today sharing a little bit of what we do at Interven. I'm a clinical data scientist in the company, and as Alex mentioned, we start discussing about how to collaborate with Neo4j when we wanted to analyze these three structures or glycans. But let me start to explain a little bit what we do. So we have heard about proteins, and these proteins, to be functional, they need to have around these sugars that Alex were mentioning. And in the cool picture at the right, you can see them in blue, right? So they are attached to the surface of the proteins. We call them saccharides or glycans or sugars. And when they are attached to the protein is able to do its job. What happened if you think about in one single protein is that you could have different glycosylation sites, and that means that the glycans are arriving there, they are attaching there. Sometimes you have them, sometimes we don't have them, but we have per protein we could have, let's say, 10 different possible sites. 3 sites, and for these 3 sites we could have like 10 or 5 or like different possibilities for glycans to be attached in there. So the complexity starts increasing because instead of looking only into one single protein, now we have to look into the same protein, consider that it has 3 sites where these sugars are arriving. And then if we consider that we have 10 possibilities, then we will be end up with 1,000 glycoforms of that single protein. So when you think about how we have been analyzing disease, at the beginning we started with the genome, then that increased to the transcriptome. When we moved to the proteome, we were already analyzing 1 million of proteins. But now when we look into the glycoforms, we are in a really, really complex level of analysis, because the detail just went a little bit higher. And it's very interesting because we know already that like some proteins are playing a really important role in different diseases, but it might be more about the sugars that are attached to these proteins, and not only the single protein as an entity. So how we do this and intervene, as Alex was mentioning already, is not an easy process. What we work with is blood samples from patients, and we analyze the serum or the plasma in these blood samples. And they go into methods that are liquid chromatography and mass spectrometry. And this is helping us to quantify the abundance of those glycopeptides. And by glycopeptides, I'm referring to the sugar or the saccharide attached to that site into the protein. So that's what we are quantifying. And this is after these methods, this goes inside to a software that was developed in-house by Intervene, and it is pretty much where all of our AI technology is applied. Because from liquid chromatography and mass spectrometry, we obtain the spectra, and this process used to be done in the past manually. Now with PIP, that is our platform, this is performed in some seconds. So it reduces the work in something. That's something that used to be done in months, now in something that it could be done in minutes. So this allows us to, instead of looking again into the proteins, we can go that level deeper. And once that we have this information about the glycopeptides in each sample of each person that is enrolled in the study, we are able to create models. We could do classifiers for, that do diagnostics for a disease in particular. We could also kind of determine if a patient is going to be or is going to respond a specific drug. We could analyze it. It doesn't have to be, we mentioned that this is about cancer, but it doesn't have to be precisely cancer. It could be another condition. And it is really important in terms of what we could do in the future if we extend this technology a little bit further. Because once that we have the information, once that we have the model, well, we are reducing that list of thousands of glycopeptides probably into a list of 20 that are representing, or that are the markers for a disease, right? And that is where the challenge arrives, because now that we have this small list of markers, then we have to look into the other information that we have about it, that it will be like the publications, the interactions with other proteins, all the information that we have about pathways, and the glycans, as Alex was saying before, the structure that they have and how this is impacting the behavior of the protein itself. So pretty much in the diagram that you have in here, it's our workflow inside the company. And this is, if you think about it, we could translate it already to how it looks into the graph, because we have the data from patients. So we have information about age, ethnicity, probably exercise. It depends, as you might imagine, per hospital and provider, the information that they give to us. But also we have that information that becomes some of the notes. Then we have different notes for the samples, because one patient could provide us more than one sample in different times. And then when we start analyzing these samples, and they go through all the methods that I just explained, it becomes more notes. And then we analyze these samples, and then we get our study notes, and that's how we just start growing and growing the graph. So at the beginning, our idea for working with Neo4j was only focusing in the structures of the glycans because they are naturally a graph. But actually, we have expanded all the data that we have. Instead of looking into a normal database, we are already implementing all this data as a graph database. This is the overall, the general schema. You could see, like in the red part at the left, we have all the samples of graph. Then we go to the glycopeptides. This is linked to the protein-protein interaction subgraph. We have also the glycans subgraph, and then we have the yellow beautiful part that is at the right, that is actually how the glycans are drawn. And at the middle I left the purple part, that is the reference. All the publications that we have could include publications or trials or studies that have been done in terms of the proteins and glycans that we are analyzing. This is not all the data that we are using, of course, is produced in-house. We are integrating other databases into our database, of course, like reshaping the data in a way that we could access easily. And these are some of the other databases that we are integrating to ours. And there are a lot of libraries for Python and Neo4j, but actually a lot of the study that we do inside Intervene from the clinical data science team is done in R. So for me, R is my everyday nowadays, and actually it's been very easy to get all the data that we have from the department, what we produce in house. Also considering all the data that comes from different databases, do data cleaning, some data formatting, writing the queries in R to produce TXT files, and then upload them into Neo4j. So I think that it's I don't know if someone else could come in the future with a better response for this, but so far this has been the pipeline that we have adopted. It has worked pretty okay, and I wanted to share with you actually how we're doing all this integration. And well, I will show you a little bit of just some examples. Then imagine that you have a protein, we have a hemo. Here is the abbreviation for this protein, and what is on the screen is Bloom. Some of you might heard that Neo4j offers Bloom and also offers the browser. This is a safe query inside Bloom, and practically what it's showing is, for hemo, that is the protein in blue, is showing what glycopeptide was identified as part of some of the studies that we have in-house, and which glycan was identified in there, and which are downregulators, or the first downregulator for this protein in particular. And then we could access also to the publication. This is a very simple schema, but it has some restrictions for you to see how easy it could be to show to someone else just by clicking or providing one name and clicking on it, just the result of different information that we already have in-house. If we go to an example for the browser, this will be a little bit different. That's how actually the browser output looks. And there is, in terms of the glycan, that is these structures, it's a world. I have to say that also when I came into the Glycoscience world, I was amazed by the amount of information that is in there, and there is a lot of publication. It's very easy to get lost when you are analyzing a protein. Then you get a glycan, then you want to know more. You have to access different databases, and it's complicated. And what I have found in here is like actually just if I want to look for publications that are specifically for my saccharides, and you can see here in the query, just giving the date, I will just pop them immediately. And if I want to put some conditions to this sugar, to this saccharide, I could provide more, and I will get this information like in less than 10 seconds. So I have found pretty useful, and I mean, we can do more queries, a little bit more complicated if we want to get a little bit more information. Um, and you can see, I think that for people like me, that sometimes we like to have the queries and the code and get the outputs straightforward, but we also like to have this visualization that actually allows you to see patterns. This is a really very useful tool. I have enjoyed it, I have to say. I've been enjoying my journey with Neo4j. I really we're looking forward to what we could do in the future and keep growing. At the moment, we are integrating more data from other databases. We are also producing more data to grow our in-house database with more examples. You could imagine that every time that we do an study, we have thousands of patients, and that all that information about the abundances of the glycoproteins in these patients goes straight to our database, and that allowed us to actually discover patterns in terms of disease and behavior. So this is what we do, and yeah, with that I will say thank you. And Alex and I think we are gonna take questions if there is some time.
Superb. Thank you very much, Karina. Super interesting project. I really like it. And I feel like you. I mean, I used to be a customer, so our highly connected data is probably the best kept in Neo4j. Yeah, it makes sense. I answered some questions already in the chat. If there are any other questions, let us know here or in the Q&A. So we will be there. And thanks again for Karina from Melbourne. So it's very late in the evening already. Thank you so much. And yeah, hope to hear from you more. And let's all hope that we can get some cool diagnosis or treatments for cancer or any other widespread disease. With Interven. Thank you very much.
Thank you. You are mute,
Karina,
Alex.
thank you very much for participating. Very nice presentation. Really loved it. And,
You know what? My nose is running, so I got really distracted. But I don't know if it's the high fever. So
okay,
sorry.
we are still online. Class
It's
safe.
okay. I
Get well
just
soon. Take a rest, and yeah,
yeah.
we'll talk to
So
each other soon. And I will stay here in the chat and answer some questions. And you
I can
enjoy your
answer
evening.
some questions if you want me to. Like at least this one that I have here. It
Oh,
says,
it's
"Do
still the
you have?"
question time,
Yeah.
probably. Okay. Is there a specific question still online? I don't see specific questions. So Ivana earlier asked if you are storing each patient as a single patient and then to it the glyco patterns, or are you storing that in terms of groups, like a patient group, like, I don't know,
No. It's everything is
individuals, right?
stored by patient and by sample.
I see.
So the abundance of glycoproteins in each of the patients vary by sex, by age.
Yeah.
So that's why it's very important to keep tracking per
Mhm.
patient of the concentration. So everything is safe individually. Yeah.
Okay, cool.
Yeah.
Cool.
And also for the quality papers, they are most of them we are taking them from PubMed. So we try to keep also the journal, right, in the journal in the abstract. That's a great area because I mean there is we tend to go always for publications in Nature, right?
I often get the question, Karina, in other perspectives, like how do you see the quality of the paper? Well, my answer is I'm not the judging person, right? We integrate and to a certain extent we trust the science, even if it's in a high-ranking journal, it can be wrong, right? We sometimes
Exactly.
measure artifacts and then you cannot really say because it's a high-ranking journal, it's more right than others, or we trust that more. I would see that rather being neutral and take a step back, integrate the data that we can get, and then have other kinds of data sources proving or disproving that this and that fact, or even have in the database. I see that often with customers having like both. Let's say if there are two scientists that have contrary approaches, to have them transparent in the database, and you can basically query both sides and say, okay, I'm trusting this more, or this experimental data supports more the other research.
Yeah, there is in terms of when we are working with data from the lab, and even when I remember when I was doing my PhD, it's the same problem because there is a lot of good publications actually in not high impact papers or journals, and there are like really high quality journals that actually accept sometimes publications that are not that good or not reproducible, at least in the lab. So it's hard to measure, and it's as you were saying, is like let's just collect it and actually like keep record of journal and everything, all the information about the publication, and then to decide if you have only one publication, probably from one regulation between proteins, and no one else have noticed ever that relationship between these two proteins. You might have some questions, probably, but what happens if never anyone else gave it a chance, and actually the relationship it is in there is just like no one has noticed. So it's a good way of discovering also, like if you think into pathways and networks in protein interaction, right, and you see that there is always a connection or maybe different paths through between two proteins, and there is one paper that is telling you that they have observed actually the direct link, you might consider it again because you might have other paths between those two proteins that are already showing you the relation. So yeah, I'll say that let's just give a chance to.
Totally agree with that. We also have a white paper with Novartis, and they integrated lots of data, and they gave it a confidence level. So they basically count the number of occurrences between two nodes and the relationship, keeping like a confidence level and saying rather of relying to an impact factor or to a high-ranking journal, saying rather how many times do I see that in my huge literature data set, and does that give me more confidence? But even then, like there can be new methods after decades, right, that can prove or disprove things. So I would keep it rather transparent there.
Yeah.
All right. Good point that a knowledge graph can also help with cross-validation or discover positive or outliers. Exactly.
Yeah.
And especially we see that with cross diseases. So sometimes we have people that are studying only cancer and they're very cancer focused, and they never saw that the same gene is also related to diabetes type 2. And then all of a sudden you have kind of comorbidities that kind of grow. And so you have new information coming to that, and this would kind of be like the disproof, right? Do you erase the cancer relationships to this protein because you have new ones in diabetes, or the other way around? Or would you keep them both? But maybe it makes sense scientifically or in the mode of action. Yes.
Yeah, exactly.
All right. Any other questions? Thanks for this very active Q&A here. I see some super engaged people like Peter and Ivana. Really cool.
I will write my email. I think that I haven't provided my email, but let me. Where can I share this? I have it in here. Well, I will type it as a question just because I have a.
Yes.
Yeah. Okay.
Very cool.
Yeah, so that's my email. So feel free to contact me if you if you have more questions or anything. But yeah, I'm very excited what we could do, and there is, I mean, there are a lot of things that when you find them, they seem obvious, but they weren't until
They
you
weren't.
see they weren't, right? So that
Exactly.
it happens to me. I love graphs, but it's every time I do a query and it's like, oh really? This is connecting this way,
Yeah.
and you don't notice, like, even if you are the designer of the graph, once that you see it, it it takes it's different, and it's a good way of exploring data, finding holes in the data, because sometimes it's the opposite. It's like that something should be connected and then they are not, and there is no way to find those gaps
Yeah.
until you visualize them. So yeah, I'm very keen, and there is no more
I think
things.
not. And I don't know if we will be kicked out here at this session, because Siddhant said that the last session is already starting. So I would say write us an email, and we're happy to take other questions via email, or just get in touch with us. Thank you very much, and bye bye.